Welcome to AP Biology. Today we're talking about cancer and the cell cycle, and we're going to talk about the most important genes involved in cell cycle control. So think of them as the brake and the gas. So first of all, there are proto-oncogenes. These are normal genes that you have that regulate the cell cycle, and they normally say go when it's time to go. So they say, hey, let's do mitosis, or let's get through a certain phase of the cell cycle. So they are positive regulators. That does not mean positive feedback. All it means is, yeah, let's do mitosis. Let's get through the G1 checkpoint, or let's get through the G2 checkpoint. So they normally activate cell division. They're either growth factor genes or something involved in the signal transduction cascade that involves growth, maybe turning on a certain cyclin or developing one. If there's a mutation in a proto-oncogene, it becomes an oncogene. So think oncologist is the doctor who studies cancer. Oncogenes are cancer-causing genes when they're mutated. So the normal one is a proto-oncogene, and if it becomes mutated, it's an oncogene. So if they're always switched on, that is one step toward cancer. So it's sort of like your accelerator being stuck in the on position. If you still have a brake, you're probably okay, but it's certainly not a good thing to always be turned on. So here's an example. This is a normal RAS gene. So here's RAS. And so the way it works is if you have growth factors, then they bind with their receptors. They'll change the shape of the receptor, the cytoplasmic domain, which will cause RAS to be activated, and it'll work with GTP, and then it will activate RAF, which will then cause a signal transduction cascade that ends up in cell division. So maybe it makes a cyclin to get you through that first phase, or maybe another cyclin to get you through the second. If you don't have a growth factor, though, then you don't turn on those receptors, you don't turn on RAS, the whole thing is off, you don't get cell division. When that's mutated, it becomes oncogenic. And so if RAS is turned on all the time, then it's saying, hey, let's do cell division. And it really doesn't matter if there's a growth factor there or not. You're kind of skipping this first step and you're going right to RAS. So that's an oncogene. So it's just an example. It's a G protein that activates cyclins. Okay, so aside from the gas, there's always also the brakes. And so there are these genes called tumor suppressor genes, and they should do exactly what they sound like they should do. They should stop tumors from forming. So they are negative regulators. That does not mean negative feedback. All it means is that they are supposed to say, hey, let's stop. We really shouldn't be doing cell division right now. So they normally inhibit cell division. If they're always switched off, that's like having no brake, so you're not turning things off, and that can cause cancer. P53 is an example, but it's a really important example, and I want you to know this one. So P53 halts division, cell division at the G1 checkpoint if it detects damaged DNA. So if there is damaged DNA, P53 will normally just stop the cell. So here's a brand new baby cell right after mitosis. It goes through G1. To get through the G1 checkpoint, you need P53 to not be saying stop. But if there's a problem in DNA and it notices some damaged DNA, it's a good idea to stop the cell before you go into synthesis and copy your DNA. So when P53 stops cell division, if there's only some damaged DNA, it just might slow it down, keep the cell in G1 for a bit, slow it down enough, activate the repair enzymes, and fix DNA. Then you can go ahead and go right through your checkpoint. Or it might just force the cell out to G0 or just to stay in G1. If there's a lot of damaged DNA, maybe apoptosis will be caused. All cancers have some type of P53 shutdown. So whether it's a mutation in the P53 gene or a mutation in something that it works on, like the P21, for example, there is some type of P53 issue in cancer cells. So here's just a visual of what a normal P53 gene would do. So here's just DNA, not the P53 gene, but just DNA somewhere. And so actually, let me just kind of talk about this a little bit. Remember how DNA makes RNA and RNA makes proteins, right? If it's the messenger RNA, that'll make a protein. So sometimes I'll be talking about the P53 gene, which would be the piece of DNA that makes the RNA that makes the protein. Sometimes I'll be talking about the P53 protein. So anyway, here's the P53 protein. if there's DNA damaged, the p53 protein will be phosphorylated, it'll be activated, and it'll go ahead and it will make a product. It'll tell another gene to turn on, which makes the p21, and it's a product that will say, hey, let's stop for a minute and repair that DNA. So if you have repair enzymes, then you can go ahead and do mitosis, and p53 allows the cell to divide. If there's so much damage that there's just too much damage to repair, the cell can then, the p53 gene will say, hey, let's do apoptosis. So p53 triggers DNA repair enzymes to go fix DNA. It also stops the cell from going into synthesis. And if there's too much DNA, p53 also says, hey, let's do apoptosis. So let's say there's a mutation in a certain cell. So let's come down here and say, here's a cell and it's got some DNA damage. Let's say, though, that the particular gene for p53 has also been damaged, and so it's making a p53 protein that's not functioning. So here's an abnormal p53 protein, and there's DNA damage somewhere else, right? And so this protein can't be activated, and so even when there's DNA damage, this p53 protein is just the wrong shape. So it can't stop the cell, and it can't turn on the DNA repair enzymes, and it can't cause the cell to go into apoptosis. And so when that happens, more mutations just occur because the p53 gene, the p53 protein's not working, and so it can't slow them down. And so you end up getting more and more mutations because you just can't repair them, and eventually the cell will become cancerous. So how specifically does p53 work? So there's a lot of things that it does, but let's look at one of them. So here is, we're at the G1 checkpoint, the very first checkpoint, and there's some damaged DNA. There are steps that will activate the p53 protein at that point. So the p53 protein is usually around, but it's inactive, and it can get broken down pretty quickly. But if it's activated, it will turn on some other DNA that will end up making a CDK inhibitor. Okay, why is that important? You might remember CDKs are kind of always hanging out, but when they have their particular cyclin, the combination of the CDK and the cyclin together, this will activate the CDK, and the CDK is a kinase, and that kinase will go ahead and phosphorylate a bunch of other enzymes to get you through that particular part of the cell cycle. Maybe the G1, you'll be getting to synthesis, so this CDK cyclin complex, the CDK part of it will phosphorylate a bunch of enzymes that will start signal transduction cascades that will turn on helicase and DNA polymerase and topoisomerase and primase, all the enzymes you need for DNA replication in synthesis. Okay, but if you have a CDK inhibitor, that stops that whole thing. So then you're not getting your cell to go past G1. So that's p53's job, or at least one of them. So here's a normal p53. So the way it works is kind of cool. So you know how DNA makes RNA and RNA makes proteins. So do you remember transcription, translation, and which one's which? DNA to RNA, it's the language of nucleotides, ATCG. This one's the language of nucleotides, ATCG. That is transcription. It's the same language. It's like me writing in English. I can do that, more or less. RNA to protein, though, totally new language. Nucleotides for RNA, amino acids for proteins. That's translation. That's what I'm really bad at. Yeah. So anything that helps a particular gene be changed or be made into RNA, that's called transcription. And we'll do a whole unit on it. But a transcription factor is a protein that binds to a particular region of DNA, causing that particular piece of DNA to be transcribed into RNA. And p53 acts as that, a transcription factor. So when there's DNA damage, p53 will get phosphorylated, it will get turned on. It binds to something called the p21 gene. When that happens, p53 will help another enzyme, RNA polymerase, to make the p21 protein. And that's what the CDK inhibitor is. That's the p21 protein. That will halt the cell at G1. It will activate DNA repair enzymes. And if it's too bad, if there's too much DNA, too many mutated DNA, mutations, I guess, too many places in DNA that are messed up, apoptosis will be triggered. So then the cells don't pass on damaged DNA, so happy day. Non-functional p53, though, if there's DNA damage, p53 can't bind to DNA because it's a protein, right? But it had a mistake in its DNA, which caused a mistake in RNA, which caused a malformed protein. That p53 protein can't bind here anymore, which means you're not going to make p21, which means you can't inhibit the CDK, which means you go right into this to the synthesis stage. You can't halt the cell. You can't get your DNA repair enzymes to get going. The unfixed damage just continues, and you can't fix even more damage, and eventually you'll probably get enough mutations to cause the cell to become cancerous. Here's probably my best picture of it. So here is DNA damage somewhere, and that is going to activate some protein kinases that will phosphorylate p53. This is the protein. And you can tell by the picture, because like here's DNA, it's double helix, right? Two strands, and this is just a globby thing. That's the p53 protein. It is inactive, and if there's no DNA damage, it's eventually going to get broken down. But if there is DNA damage, p53 will get phosphorylated. Now it's active. When it's active, it acts as a transcription factor, which means it goes to the DNA of a particular gene. It goes to the p21 gene, causes transcription to happen. Now you have a p21 mRNA, which can make a p21 protein, which happens to be a CDK inhibitor. So this cyclin CDK complex should go ahead and start a signal transduction cascade that makes all of the enzymes become activated, all those enzymes for DNA replication. But because you have this C21, it just halts that whole thing. So now the CDK is not active, and it can't go through. It can't go ahead and activate get into the next part of the cell cycle. So last slide here, cancer develops after 6 key mutations. Unlimited growth, that means oncogenes, proto-oncogenes have been turned into oncogenes. So you are turning on either a growth factor or like maybe RAS, one of the parts of the signal transduction cascade to make a cyclin, for example. You are ignoring checkpoints. So p53 is messed up and can't stop you from moving from one part of the cell cycle to the next. You don't do apoptosis. So if you have that much DNA damage, you should die, but you don't if you're a cell. Immortality. So telomerase is active. Promotes blood vessel growth. So angiogenesis, you're bringing blood vessels with their nutrients and their oxygen toward the tumor so it can grow faster. And then finally, they don't respond to density dependence anymore, density inhibition anymore, and they don't care about having anything under them. They will just grow. So something about those CAM proteins are messed up. And those are the touch sensors that are off. I guess I'll do this last one. What causes mutations can be lots of things. Can be UV radiation, can be chemical exposure, maybe at work, can be radiation exposure, again, maybe at work. Cigarette smoke, not only does that bring pollutants into your lungs, but it also brings tar, which can hold those pollutants in your lungs longer. Pollution, if you live where there's a lot of pollution, and age, kind of well, it's all a bummer, right? Genetics, maybe your immune system isn't as good at killing cancer cells, or maybe your enzymes for DNA repair are a little slower, or maybe you're born with one of the mutations that you need in order to develop cancer already. Maybe not all of them, but one of them, right? Maybe there's quantum weirdness, which is causing some T's to become A's or C's to become T's or something. All right, bye.